for coming after this
Hi.
wonderful introduction. I guess let's hop into it. So,
Welcome.
neotropical poison frogs have always held a special place in my heart. They exhibit a wide diversity of mating systems, parental care strategies, toxic regimes, phenotypes, and speciation events. And because of this diversity, I became
Voy a
broadly
estar allá afuera,
interested
ya
in the
llegó en...
evolutionary relationship of these frogs, especially since there's still a lot of uncertainty involving these relationships. So that leaves much to be disentangled. So although we are seeing with an increase of the use of genomic data, we have begun to understand some of these connections. And so this can be seen with the recent studies regarding genus Ammariga that currently includes 31 described species. There's a little bit of a spoiler alert here. So among these described species, one group I'm really excited about is this Ammariga bacilli species group, or hereafter I'm going to refer to them as the ABSG. So get used to
I'm sorry.
hearing ABSG.
Uh,
In recent broad
the girl,
studies
she
of the
was,
genus
yes.
Ammariga, the ABSG was
No,
teased
no, it's
apart,
okay. It's
consisting
okay.
of
She's
three
in Laura's
morphological
office.
variable species,
So
Ammariga
it's fine.
bacilli, Ammariga pepperi, and Ammariga yoshina. And so this is
I'm going
where
to
I
be
began
outside.
to focus my dissertation work. Okay, so generally, the genus Ammariga are large aposematic frogs that are restricted to montane habitats. So specifically, the distribution of the ABSG extends along the Andean foothills throughout north-central Peru. And as you saw from my introduction, I hiked quite a bit in that. So the ABSG are each geographically distinct, and I want you to notice the difference in color pattern represented with this graphic. So to better illustrate this phenotypic variation, each species in the complex possesses a unique combination of bright dorsal coloration, varying in different degrees and combinations of greens, yellows, and reds amongst the species. In addition, calls can be differentiated between the species by the call rate, the silence between notes, and the dominant frequency. And the graphic represents these comparisons. So you can see the frequency and then the silence between notes, and then you can see how often these sounds spike. So these differences between species calls might be easier to understand if you can hear them. So bear with me for a second because I have to adjust this just a tiny bit so I don't blast your ears out. Oops. Okay, so this is Ammariga yoshina. This is Ammariga pepperi. Again. Oh yeah, this one I need to be loud. Sorry, this one's the quietest of the calls. And then we have Ammariga bacilli, which this one I think I need to turn down as well. So hopefully we can see, we could hear that difference. So Ammariga yoshina has the fastest call rate, the shortest duration of silence between notes, and the highest dominant frequency. Ammariga pepperi has the slowest call rate, the longest duration of silence between notes, and an intermediate dominant frequency. And then we have Ammariga bacilli, which has an intermediate call rate and duration of silence between notes and the lowest dominant frequency. And so all of these aforementioned components provide evidence of reproductive isolation. So once again, now that we know what we're looking for, I'm going to make you listen to the calls one more time because why not? We're at a frog talk. So I don't blast your ears. Fastest call rate. Then we have the slowest call rate, which is the pepperi. And then we have bacillary. Cool. We got to listen to frogs today, y'all. All right. So in 2009, Brown and Toomey described two of these species, Peppery and Yoshina, because of their acoustic differences, and did so because in poison frogs in particular, research has shown that male advertisement call strongly influences reproductive isolation between species. So under this assumption, based on the acoustic data, they described Peppery and Yoshina. So despite the acoustic evidence, however, we see several instances of non-monophyly with Yoshina indicated in the yellow throughout the tree, which often violates standard phylogenetic species hypotheses. And so now we're left with this conflict between the acoustic data and this incredibly messy genetic data, which led me to needing to further disentangle this mess to better understand these evolutionary relationships of this group. So the central aim of my dissertation research is to provide context into the mechanisms of speciation and reinforcement in the Amarega bacillary species group. And so this consists of several objectives. And so I'm going to answer this question by providing taxonomic clarification, by conducting the most comprehensive sampling to date, and by performing genome-wide nuclear sequencing using ultra-conserved elements and mitochondrial genomes. And I'm going to reassess species boundaries using a robust male acoustic data set. So I will also be performing multiple population genetic and phylogenetic analyses by estimating gene flow and testing for integration among species. And then to give further context into the evolutionary history of the ABSG, I will estimate times of divergence. And then finally, I will determine the roles of climate dynamism and geographical boundaries and how they relate to the flow of genes across the landscape, and evaluate how key environmental variables and paleoclimatic periods shaped their biogeography. And so this brings me to my first order of business in tackling this amazing mess that is the ABSG. And so you saw some of these photos already. But while I'm not just showing you my vacation photos, what became overwhelmingly clear was that I needed to conduct more robust sampling, both genetic and acoustics, in order to begin answering all of my questions. So I sampled throughout known ranges of the species group, while also adding additional localities from areas closer to the edge of their distribution. I do just for fun. This guy I saw every day. I hiked every day 7 miles and I saw this guy every day. Same spot. It was like fate. So after my robust sampling, the first thing I did to clarify taxonomic relationships in the ABSG was to conduct a maximum likelihood analysis using nuclear ultra-conserved elements, or UCEs, in IQ tree. So when I say UCE or nDNA or nuclear data, I'm talking about the same thing for this talk, so I might use them interchangeably. And so this was where the first of many head-exploding discoveries happened. From this tree, according to Brown and Toomey in 2009, what was thought to be bacillary is now polyphyletic. So it's sister to Yoshina, which follows classification from Guillory et al. In 2020. And then we have a second bacillary, which is sister to Peppery. And so to better illustrate this geographically, I updated the previous occurrence distribution map. And so at this point, it's worth noting that the holotype for bacillary is found where it's indicated with the star, which is here. And I need a... I'm too scared of knocking this over to put that up. Okay, it's also noteworthy that the yellow bacillary individuals here were previously thought to be from the type locality of the true bacillary species and was in agreement with previous work from Guillory et al. In 2020 that places Yoshina and bacillary as sister to Peppery. while the green labeled group also contained individuals from bacillary populations and was thought to also contain the holotype of the true bacillary. So now I have, who is the true bacillary? So for the last few decades, it has been unclear which morph of this frog represented the true holotype for Ammariga bacillari. And for anyone unfamiliar, there can only be one. So this confusion has spanned the last several decades of research regarding the ABSG. So to provide better clarity for these errors, I delved into old literature to ascertain the true identity of bacillari as originally described by Mellon in 1945. And so I found the following quote of the described holotype to be light green with large dots. Furthermore, I found a painting and holotype sample of bacillari as Mellon described, so which is here and then here. So it's safe to conclude that this is the true bacillari. I also found reference to the yellow morph from Silverstone in 1976, where he concludes that the yellow morph is likely a new species. So it seems like I'm on the right track here. I also found picture evidence of the yellow morph occurring near Roque population, which is the problematic population from Rainier Schulte. And so given this literature review to clarify true species identity and the phylogenetic information, the next step was to assess species boundaries using calls to better rectify the inconsistencies of previous work. So using Raven Pro and Audacity to clean and prepare calls, it was found that calls among all lineages are statistically unique. I'm not going to make you listen to the calls again. I'm sorry. But here I'm showing another figure for more clarity. And the combination of the length of notes and spacing between the notes suggests that they have diverged and suggest reproductive isolation. So now that I have both phylogenetic and acoustic data supporting that I have a new species, I can now present Ammariga currienti with this publication, actually as of last night. And so a little fun fact, the name comes from the Quechua language, which is an indigenous language spoken in the area the frog is found, and it directly translates to golden sun for its golden color and for the behavior of calling as the sun rises and sets. And so using this updated taxonomy, my phylogeny provides much needed clarification to the ABSG. So given this new taxonomy, I wanted to further assess evolutionary relationships by analyzing both nuclear and mitochondrial genomes. So to look at different evolutionary depths, I obtained two concatenated maximum likelihood trees, one from a nuclear UCE dataset and one from a mitochondrial dataset across the genome. So I was dealing with a fairly large dataset for both trees here. And the other thing I want to draw your attention to is that the results of the nuclear genomic tree indicate each species is reciprocally monophyletic, while the mitogenome tree shows multiple instances of mitochondrial capture highlighted by the red lines and represent historic hybridization in the ABSG. This is because mitochondrial DNA is maternally inherited and non-recombining, so it can reveal the inheritance of genetic signatures, in this case from other species, giving us a glimpse into the historical admixture of the complex, while the nuclear genomic data shows us the contemporary version of our complex. And so this leads to the next head-exploding discovery, because in a perfect world, both of these trees would match topologies. So I wanted to know why I was seeing this huge incongruence between the nuclear and the mitogenome. And so there are a few consequences of hybridization, and our evidence show that we have quite a bit. So just to be clear, while I'm showing you these graphics with my frogs to better understand this, I am not necessarily saying that these outcomes is what is happening with ABSG. I just wanted to provide a graphical representation for outcomes of hybridization. And so some of these outcomes include reinforcement, which is the evolution of prezygotic mechanisms in areas of overlap or hybridization. as a response to selection against hybridization. Then we have the establishment of hybrid zones, which is areas where genetically distinct populations interbreed. They produce hybrids that will either lead to speciation or the fusion of the two taxa. However, these areas can remain stable over a long period of time due to a balance between dispersal of parental taxa into the zone and selection against hybrids. Then we have species reversal, which occurs when isolating mechanisms are incomplete, and the continued interbreeding and backcrossing causes the genomes of the two species to become so thoroughly mixed that they're no longer genetically distinct, and they collapse into a single species. Finally, we have hybrid speciation, where this graphic very broadly represents a hybrid speciation event where two lineages that have contact result in gene flow, leads to the hybridization between the species, and then for whatever reason, there's a separation that leaves the now isolated hybrid lineage to become a separate species. So these hybridization outcomes depend on population demographics. So understanding current structure and admixture is key to predicting if reinforcement will stabilize species boundaries and if a hybrid zone will persist or other dynamics like reversal or speciation will emerge. So I used a discriminant analysis of principal components to essentially group genetically similar individuals together. So from this analysis, 5 populations were recovered. And to better illustrate these findings, I overlaid the results on the previous occurrence distribution map, where the numbers and colors represent the DAPC results. And this division of Yoshina, so whenever I say I have 5 population, it occurs here with Yoshina and is seen here as well. So this division makes sense geographically because these two populations are separated by a huge river, which is illustrated by this blue squiggly. So to further assess admixture, I used NGS admix, which estimates individual admixture proportions from genotype likelihoods, detecting recent structure from events recent enough to cause allele frequency differences. So it turns out K equals 4 was the optimal K for NGS admix, showing residual admixture between Currienty and Bacillary and Peppery. And it is worth to note that K equals 5 was the second highest supported. So given the genetic inconsistencies between the UCE and mitogenome and the population structure, I wanted to further investigate gene flow between species. So DAPC and NGS admix depict recent signatures of gene flow indicated by the red X's here. And I wanted to look at deeper evolutionary depths. So to infer deep signatures of hybridization, I implemented a genome scale calculation of D and F4 ratio statistics, which is used to assess evidence of gene flow between populations or closely related species. And it's more sensitive to integration occurring over long, extended evolutionary timescales using my nDNA data or my UCE data. And so as I move through this graphic, percentages and red arrows here represent alleles shared, and it reveals with this analysis that we have reciprocal gene flow between Currienty and Bacillary with approximately 17% of alleles shared. We also have gene flow from Currienty into Peppery with approximately 14% of alleles shared. And again, I wanted to better visualize in comparison what we're seeing with the mitogenome. So the black dashed arrows represent our mitogenome results that show admixture between Bacillary and Peppery, admixture between Currienty and Bacillary, and admixture between Yoshina and Currienty. So again, to put this all together, and just as a reminder, the red arrows are alleles shared, which are the results from my D-suite or my nDNA. And then the black dashed arrows are from my mitocapture from my mitogenome. And so this is what it kind of looks like all together. So just for a quick recap, at this point, we're seeing further evidence of 4 species in this group. We're also seeing several lines of evidence of admixture, particularly between these 3 species. But I still didn't successfully resolve the incongruence between the mitogenome and my nuclear UCE phylogenies. So this leads me down a rabbit hole to find additional explanations for what's going on. So I bring you back to the possible outcomes of hybridization that we discussed before. And so how we can start to disregard these possibilities with the ABSG. So again, we have reinforcement, which is the evolution of prezygotic mechanisms in response to hybridization. But based on the mitochondrial capture and the nDNA and mitogenome incongruence and the contemporary presence of bacillary, we can disregard this possibility as an outcome for the ABSG, though reinforcement likely did occur post-speciation. And then we have hybrid zones where distinct populations hybridized, balancing dispersal and hybrid selection to persist long-term with outcomes of speciation or fusion. So we don't see stable hybrid zones in the field, and this is also reflected in the genetic data because we have nDNA and mitogenome incongruence with mitocapture and the existence of bacillary, so we can also rule this one out. And then with species reversal, we have a breakdown of reproductively isolating mechanisms to the point where 2 parental species are replaced with a hybrid swarm. So once again, because we have bacillary, curianti, and peppery, we can go ahead and disregard this outcome. So now finally, we have a hybrid speciation event, which occurs when 2 lineages come into contact and exchange genes, forming a hybrid lineage that later becomes isolated and ultimately evolves into a separate species. So there are a few cases of this process in mammals, birds, fish, and amphibians, and based on the results showing gene flow between lineages and the discordance between the mitogenome and nuclear DNA, I begin to think that we might have a good case for this with ABSG. So, well, I just threw something huge at you. So up until this point, what specific evidence do we have to support this hybrid speciation hypothesis? So first, once again, you're probably going to hear me say this a lot. We have a scenario with our nDNA tree and our mitogenome tree that shows the contemporary representation of our complex and the historical representation. So one thing I want to do first is to make a slight alteration of the mitotree to better illustrate this issue. So again, I am not altering this for any analyses. I'm just doing this for graphic to better explain kind of the issue that I have going on here. So I'm manually removing this branch because I'm not focused on this specific topology, which is likely the result of more recent admixture. I want to focus on this relationship that is wildly incongruent to the UCE relationship. So my big question here is, why do curianti and bacillary share an ancestor in the mitogenome? And so the leading explanation is that at some point in time, the ancestors of curianti and peppery hybridized. And this is represented by the dashed line and red X's. And so this gave rise to the bacillary lineage, so that bacillary retains the curianti maternally inherited mitogenome, which is illustrated by this relationship. And so with that main issue squared away, the other lines of evidence for this scenario are, I found further evidence of historic gene flow between the species using the D statistic and F4 ratio statistic, which represents the percentage of alleles shared. One thing that the D statistic tests for is incomplete lineage sortage and admixture by using the ABA-BABA framework to detect a significant deviation from 0. And so this indicates patterns that are attributed to admixture, which we find here. Finally, we see acoustic divergence, where we would expect that the hybrid species, so in this case, bacillary, should be intermediate to parent populations, so curianti and peppery, which we do in fact see here with these highlighted call parameters marked with the red arrows. So based on these qualitative results, I wanted to quantify our hybrid speciation hypothesis. So to do that, I wanted to see if our genomic data had genetic signatures of hybrid speciation. So we ran approximate Bayesian analyses. So in brief, this method is used to simulate data among different evolutionary scenarios and then assess which scenario best matches our observed genetic data. So in our analysis, we compared two scenarios, one following the normal circumstance of speciation and the other following a scenario of hybrid speciation, which is illustrated here. And surprisingly, we found decent support for the alternative hypothesis, which represents hybrid speciation with a posterior probability of 57%, or approximate 57%. So given all of these results, we have support that bacillary was the result of hybrid speciation between the ancestors of Currienti and Peppery. So I have evidence of deep historical integration. So what ecological processes facilitated these patterns? So I first needed to look at the time of divergence for the ABSG, since they are contemporarily unique. And so to calculate divergence time, I used a GTR model in real time using the maximum likelihood topology previously created in IQ tree as a reference. And so since there are no known fossil records for dendrobatids, I used the estimates from Guillory et al. And calibrated the ancestral node of the tree to a uniform distribution. And so the important takeaway here is that the crown age for the ABSG is approximately 2.5 million years ago. So finally, I will determine how the roles of climate dynamism and geographical boundaries relate to the flow of genes across the landscape and evaluate how key environmental variables and paleoclimatic periods shape their biogeography. So I used the 19 standard bioclimatic variables at 1 kilometer. I used soil layers and several topographic layers. So these layers were downscaled to a resolution of 30 meters using the R package Matchy spline. And so the cool thing here is that each map of the species distribution is divided into 3 by 3 squares, which better illustrates how each species occupies different areas and further aligns with the initial thinking about their distinct geographic locations. And so to clarify, each map is a one-to-one comparison. And so to estimate whether the ABSG have evolved different niches, each species was mapped using the downscaled environmental layers that contributed to 5% or more to the model predictions. And so the occurrence environmental data was loaded into the R package Humboldt, and paired comparisons were iteratively compared to evaluate the environmental space occupied by each species. And so we found that all niche overlap tests and divergent tests resulted in significant equivalence tests and can be concluded that all species comparisons occupy different niches and have diverged their Grignelian niches. And so to test how key environmental variables and paleoclimatic periods influence their biogeography, I used resistance GA to create mixed spatial models of landscape genetic diversity. So this program uses resistance layers, which is just a raster map where each cell has a value representing how hard that section of landscape is to move through. So low values is easy movement, high connectivity. High values is hard movement, low connectivity. So I generated 18 resistance surfaces at 5 kilometer resolution. Each of these served as a separate hypothesis. And the combination of all of these resulted in 366 models. Resistance GA uses a genetic algorithm implementing pairwise genetic data and resistance distances to calculate resistance values. And so for this, we use commute distance and least cost path. And so these were performed for every pair of the 18 spatial hypotheses, as well as a single predictor. So to put that into perspective, that was 18 spatial hypotheses for a total of 366 combinations for each of the two different models, commute distance and least cost path. So this takes several months to complete, and to say that this was not a real fear would be an understatement. This computer screamed at us for days. So from that, the best model predicted based on AICC was the least cost path composite surface of the last glacial maximum and clay. There were also two top commute distance models based on the AICC, which was a univariate model using elevation and a univariate model based on current climate. And so just looking at these different maps, the 5-kilometer least cost path composite layer of the last glacial maximum and clay, we can see that on the left, the landscape friction where like the warmer colors indicate higher friction, and the map on the right, so right here, shows where these corridors of connectivity through resistance surfaces where the warmer colors indicate high connectivity. And so again, we have the univariate 5-kilometer models for elevation and current climate. So we have elevation and we have current climate. So again, this follows the similar thing as the last maps, where the left map shows the resistance surfaces with warmer colors indicating higher friction, and the right hand side of the map show areas of connectivity through these resistance surfaces with warmer colors as higher connectivity. Here we have those beautiful maps to take in that took months to make. So elevation emerged as an important resistance factor, explaining genetic distances in our resistance GA models for the species group as a whole, implying that movement and gene flow are constrained along elevational gradients. So post hoc comparisons of species elevation distributions mirror this pattern, where Bacillary and Currienti occupy higher elevation sites, Yoshina has lower elevation sites, and Peppery is intermediate, suggesting that species-specific elevational niches contribute to the elevational-driven connectivity pattern detected at group level. So, okay, what did I accomplish throughout my whole PhD? So through comprehensive phylogenetic and morphological analyses, I elevated the yellow morph that was once considered Bacillary to Currienti. And so with this updated taxonomy, we now have well-supported monophyletic clades. I named a species, and so I just have to show you again. Which also means that I increased the number of described Amarega from 30 to 31. So that was a little spoiler alert from the beginning. After several genome-scale population and phylogenetic analyses, Bacillary likely originated through the hybrid speciation between ancestral populations of Currienti and Peppery. And so this pre-zygotic reinforcement breakdown was likely caused by the promiscuity of the ancestor to Currienti, which is seen with the incongruence between the nuclear and the mitogenome. Landscape genetic analyses and spatial configuration of the Andean topography and regional climatic variation have influenced patterns of gene flow and divergence in the ABSG. So in other words, the physical and climatic landscape of the Andes has promoted the ABSG's diversity. Results further indicate that ecological specialization and geographic barriers have contributed to diversification. So each species occupies regions of environmental space consistent with adaptive ecological divergence shaped by heterogeneous mountain environments. And this is particularly important because adaptation to different environments reduces gene flow, promotes reproductive isolation, generates and maintains biodiversity across these heterogeneous landscapes. And then despite evidence of historical integration, pre-zygotic barriers such as ecological divergence and sexual selection reinforced isolation and stabilized distinct species. So it is likely that these reproductively isolating mechanisms, which are the acoustic differences and the ecological niche differences, occurred secondarily post-hybridization. So an important aspect, so now we're at the end. So an important aspect that is obviously missing from this story is understanding how mate choice affects these strong signals we're seeing in reproductive isolation and how larval interactions might shape populations. So in the interest of further investigating this missing component, and because I just can't help myself, one of my many side projects. I am investigating mate choice through both acoustic and visual trials. Unfortunately, Yoshina is missing from this graphic. So I am also compiling larval natural history by taking loads of morphological measurements and investigating larval interaction among and between species through competition trials. And so this was one of the videos Jason was trying to show where the tadpoles are moving and eating from the surface. So with that, I wanted to acknowledge my funding sources. Without this, I would not be able to have supported myself. So thank you. I also wanted to thank my committee for providing assistance and for challenging me to be a better researcher, and for hopefully, not to jinx this, putting the doctor in front of my name. I want to thank my master's advisor, Kyle Summers, for taking the chance on me in the first place so that I could be here today. I also wanted to thank the graduate students in the department who provided support and epic Hobbit-themed get-togethers, and the countless undergrads who have assisted in the field and with projects. I also realized as I was putting these slides together, I have a bad habit of not taking enough pictures. And I also have the habit of making about the same dumb face. So if I forget your picture up here, it was not on purpose. You have influenced me. Oh boy, here we go. I wanted to give a special acknowledgement to Jason Brown. He specifically brought me back here with his infectious passion for research and for these frogs. He challenged me to think outside of the box and to broaden my scope of thinking. He supported me through all of my doubts and made me a more rounded scientist. And so most importantly, he taught me to be fearless and take chances. So thank you. To all of my friends I made along the way, without you, I wouldn't have made it through with even the smallest percent of sanity I did end up with. I don't even have enough words or even time to express how much you truly mean to me, and hopefully you know that. Finally, I wanted to dedicate this to my family. Everything I've done for you. You've been my greatest strength, my greatest support, my greatest sense of comfort, and the greatest fans of my life. I'm so thankful to have had you along my journey of life. I do wish that all of us could have seen the finish line. With that, any questions?